Okay, let's go ahead and finish up this section on spores. So, as you do spore staining in the laboratory, you're only doing one particular species, so you're not going to get an appreciation for this in the laboratory. But you can do somewhat diagnostics with spore-forming bacteria, because certain bacteria that form spores put them in different places. Like, for instance, the bacterium that causes tetanus, Clostridium tetani, forms endospores at the very end. So they kind of look like a miniature tennis racket, or like an old school tennis racket. Whereas other ones like Bacillocellus form them in the center. It's kind of a minor point, but just kind of to show you the variation that exists in the pattern of sporulation. Some of the more interesting stuff about sporulation is how long can these spores last, which is actually pretty controversial. We know easily that in nature, spores in the soil can live 50 or 60 years. Other ones, people have found them documented from archaeological sites more than 2,000 years old. It gets a little bit more controversial. You can get in a lot of arguments when you go much older than that. So kind of the Jurassic Park story, right, where somebody was able to isolate dinosaur DNA from a mosquito trapped in amber. That idea for that Steven Spielberg epic, I think that was a Spielberg, but anyway, that epic movie kind of comes from the fact that people were actually able to isolate spores from bees trapped in amber and resuscitate them 25 to 40 million years old. There's always controversy because in the laboratory, these bacteria exist all over the place anyway. So how do you tell a 25 million year old bacterium from a bacterium that's in your laboratory today if they're the same species? But I think most people do tend to accept the fact that you can isolate spores under certain very special conditions that are millions of years old. So it's actually kind of a neat thing, but it's a testimony to how resilient these endospores are. Remember that they have evolved to allow the organism to survive prolonged periods of difficult times. They are not designed to allow bacteria to be spread. So that's a different type of spore that we're not going to talk about. Now, part of the reason why it's interesting to talk about spores is because they actually have a major role in infectious diseases, especially in hospital-acquired or what's known as nosocomial infections. So you've probably all heard of the bacterium Clostridium difficile, oftentimes just referred to simply as C. diff. This is a bacterium that can be a normal component of our intestinal tract. Probably about 5% of us carry this bacterium at low levels in our colons, and it will never cause us disease unless you're actually treated with a large amount of antibiotics that kill the other bacteria but not the C. diff. Then without the competition, it can grow out. But this is what C. diff looks like with a spore near the terminal end of this bacterium. Now, this is particularly a problem in hospitals. So this is a major cause for morbidity and mortality in hospitals. Because you think about the number of people who are inpatient in hospitals who have been treated with antibiotics for various reasons. Their intestinal tracts get somewhat depleted of the normal microbes that are in there. So that ecosystem is depleted in some manner. Now, if that person with a reduced colonic microbiota or bacteria ingests these spores of C. difficile, they're much more likely to come down with what's known as pseudomembranous colitis, which is a highly lethal form of the disease. These people are already sick, so they don't have a good defense system, perhaps. And the spores from C. difficile, so you're seeing in the laboratory how difficult it is to visualize spores. You literally have to cook the bacteria in order to get the dye into the spores. Well, in the hospital, all of the infection control is done, all of the disinfection is done, is all designed on killing vegetative bacteria. There are very few bactericidal, if any, chemicals that you can use on your hands that are going to kill spores. So what's happened is that now C. diff has actually evolved to be a hypersporulating organism. As part of its evolution, it now it forms spores much easier. So surfaces in the hospital can very easily be contaminated with spores that no matter how well you wash your hands, or you're likely to carry them into the next week. The spores can be ingested, and these spores are highly evolved to live in our intestinal tract because they're not even killed by the acid in our stomach. We think of our stomachs as means to digest food, and they certainly help us to digest food. But one of the other really critical roles of the human stomach is to help protect the intestinal tract against infections. Most parasites, bacteria, and viruses that we ingest are destroyed by the acid in our stomach. Only a few manage to get through. This spore is very resistant to the acid in the stomach. And it's actually evolved such a tight association with the human host that what stimulates that spore to germinate, how that spore senses that it's home, is that it senses bile acids. So once the spore has made the trip safely through the stomach and into the small intestine, that's the first place where it encounters bile acids being secreted via your gallbladder, coming originally from your liver. And now it realizes, hey, I'm home. It's time to germinate. So those spores will germinate into vegetative bacteria away from the toxic effects of the acid, and they can proliferate to huge numbers if they don't have competition. And when they do that, they'll release a large amount of toxin, which will cause necrosis or cell death, especially in the colon. So they form this really lethal pseudomembranous colitis in the lumen that in minor cases will cause diarrhea, but in severe cases can actually be lethal. And people are infected for life with this, or they can be infected for life. We're going to come back and talk about this later on in the class when we're actually talking about disease mechanisms, because the disease mechanisms that go with this are fascinating. But this is just my attempt to show you one of the reasons why it's important that you understand something about spore formation and its difficulty. So here's
certain gram-positive genera, including some
an animation without me talking for a while.
Heat and desiccation-resistant spores. Most of our knowledge of bacterial sporulation comes from the gram-positive soil bacterium, Bacillus subtilis. When growing in rich media, this microbe undergoes normal vegetative growth. B. subtilis can replicate every 30 to 60 minutes. In contrast, starvation initiates sporulation, an elaborate 8-hour genetic program that ultimately yields a spore. Sporulation can be divided into 8 morphological stages. Stage 0 represents the point at which the vegetative cell decides to use one of 2 potential polar division sites to begin septum formation, instead of the central division site used for vegetative growth. Stage 1 involves replicating and stretching the DNA into a long axial filament that spans the length of the cell. Ultimately, one of the polar division sites wins out, and in stage 2, septation occurs, dividing the cell into 2 unequal compartments: the forespore, which will ultimately become the spore, and the larger mother cell from which it's derived. Each compartment contains a chromosome. In stage 3 of sporulation, the mother cell membrane engulfs the forespore, resulting in a structure encircled by 2 membranes. In stage 4, the mother cell chromosome is destroyed, and a cortex, made up of a thick peptidoglycan layer, is placed between the 2 membranes surrounding the forespore protoplast. This cortex will be important in providing heat resistance and maintaining dormancy to the spore. In stage 5, layers of coproteins are deposited on the outer membrane. Stage 6 completes the development of spore resistance to heat and chemical insults. This last stage includes the synthesis of dipicolinic acid in the mother cell and its uptake into the core of the spore. Dipicolinic acid is important in maintaining dormancy and in promoting spore dehydration, a characteristic that makes the spore heat resistant. In stage 7, the mother cell, now called a sporangium, releases the mature spore. Spores are resistant to many environmental stresses that would kill vegetative cells. The nature of this resistance is due in part to a spores having only 10 to 30% of a vegetative cell's water content. But spores also contain small acid-soluble proteins that bind to and protect DNA from damage by ultraviolet light and various toxic chemicals. Fully mature spores can exist in soil for at least 50 to 100 years and have been known to last thousands of years. Once proper nutrient conditions arise, another genetic program called germination is triggered to wake the dormant cell, dissolve the spore cortex and coat, and release a rehydrated, viable vegetative cell.
So, a few minutes without me talking. As an aside, so if you haven't noticed, I put another folder in the Blackboard site. So these animations, including this one, that I embed into the PowerPoints, are now available for you to look. So as you start to study for the exam, to go over this. This is an especially good animation because it explains the formation of the spores much better than I do. I am not interested, I will never ask a question like what happens in the fourth stage of sporulation. What I am interested in is that you have a general appreciation for how the process works. Okay, so now let's move on to the next section, which is going to be about microbial nutrition and metabolism. And so just to start off, because remembering that one of my major objectives in this class is to really show you what diversity is in life, because microbes really have all of the genetic and biochemical diversity, much more so than all of the organisms that you know and love. And so I stole this quote from a previous textbook that we used years ago that I think really captures something about this. So it is a most remarkable nutritional characteristic of microorganisms is their extraordinary flexibility with respect to carbon sources. There is no naturally occurring organic molecule that cannot be used by some microorganism someplace, which is really a powerful statement because if you think about the number of pollutants that we have in the world today, many of them are organic. Things like petroleum products, insecticides, all of these products, some of which are not even naturally organic. Generally, someplace there is a bacterium that can break them down, use them as a carbon and energy source. So in terms of remediating polluted sites, understanding microbial metabolism is crucial because what a lot of people actually do is they mine for bacteria that can break down various pollutants and then can use those to help clean up sites. So they're invaluable in bioremediation. And oftentimes these bacteria, even though there may not be one bacterial species that can break down a toxin or an insecticide, there's usually a guild or a collection that can do it in a process known as syntrophy, where you have, in this very simple example, this hypothetical example, you have toxin A in the environment. Bacterium A can break down toxin A into toxin B. So it's still a toxic molecule. And like most end products of metabolism, if toxin B builds up to a high concentration, it inhibits bacterium A. Nobody likes to live in their own waste, including us. So bacteria, as they break down and secrete a waste product, would actually be toxic, it would inhibit this. But oftentimes what happens is that you have a second, so in this very simple cascade, bacterium B can take toxin B and break it down into a non-toxic substance, which eliminates toxin B, which allows bacterium A to still metabolize and to get rid of more toxin A. So oftentimes these are multi-species consortia that help to break down these pollutants. So here's a more real-world example, a very well-studied example of syntrophy between an anaerobic soil bacterial genus called Syntrophus that has the ability to hydrolyze benzoate, so this benzene type structure. By itself, under anaerobic conditions, so in other words, no oxygen present, it can break benzoate down into cyclohexane carboxylate, shown here. Again, I'm not an organic chemist or any kind of a chemist, biochemist or whatever. I'm not interested in you understanding the chemistry. The point is that by itself, Syntrophus can go no further than this cyclohexane carboxylate. Now, the way that it actually finishes the job and breaks it down is by entering into syntrophic interactions with methanogenic archaea. We're going to talk a lot about methanogenesis and the archaea because it's really important for greenhouse gas production, but it's also very important for the proper function of many ecosystems. But in this particular case, these are archaea that have the ability to take the waste products that Syntrophus has generated to this point. CO2, especially important is hydrogen. Hydrogen gas, as a byproduct of many bacterial fermentations, is inhibitory to that continued fermentation. If the hydrogen gas in that environment reaches, like, say, 1 atmosphere of pressure, that stops the fermentation, in this case of benzoate to cyclohexane carboxylate. That's the reason why, by itself, Syntrophus can't complete the breakdown of cyclohexane, because it's inhibited by, especially by hydrogen gas, but also by its other byproducts like formic acid, CO2, and acetate. Now, if you add these wonderful methane-producing archaea, what they can do is, by removing hydrogen gas and removing CO2 to generate methane and more CO2, they get rid of these inhibitory molecules, which allows Syntrophus to break this down further, so that basically all of the cyclohexane carboxylate will now be broken down, metabolized into formic acid, hydrogen, acetic acid, which is actually also used because it can inhibit it. So again, this is why this is such a well-studied system, is because it's such a simple system. It just really relies on one bacterial species and 2 members of the archaea. So this is an example of syntrophy, or sometimes referred to as cross-feeding. So these bacteria are being cross-fed by a product from the first bacterium. I prefer the term syntrophy to cross-feeding, but the literature oftentimes calls it cross-feeding. Now, in terms of microbial nutrition, all microbes are broken down, so we are humans, and we like to break things down dichotomously and pigeonhole things, and this is what we do. And you can break down bacteria, all of them, well, all organisms on Earth for that matter, based on their source of carbon. Where do we get our carbon from? Where do we get our energy from? And where do the electrons that we use come from? So in terms of carbon sources, there are 2 flavors of microbes, or 2 flavors of any organism. There are autotrophs, and we've talked a little bit about those. We talked about the carboxysome and the Calvin cycle and all of that stuff in terms of cyanobacteria. In autotrophs, carbon dioxide is the sole, or perhaps only but at least the principal form of organic, or carbon, and it's being formed into organics. We, on the other hand, fall into the heterotrophs in terms of our carbon sources. And most of the bacteria that you will ever deal with for the rest of your careers, unless you become microbial ecologists for some reason, or industrial microbiologists, most of those microbes will be heterotrophs. So these are things like us that use reduced preformed organic molecules, just like the breakfast that we're degrading right now. Most microbes use glucose or amino acids or fats for their nutrition. In terms of energy sources, again, very simple. Energy is either coming from light, in which case it would be a phototroph, troph meaning to eat, right? So this literally means to eat light, as opposed to us and most of the bacteria that you'll ever deal with for the rest of your lives are chemotrophs, where the energy source comes from oxidizing some type of organic or an inorganic molecule. It simply means that that light is not the energy source, that it's coming from a chemical. Chemotroph does not differentiate between whether that energy source is organic or inorganic. It simply means that it's coming from the oxidation of a chemical product. And then the third concept that these microbes are broken down into is based on the electron sources. So remember, when we oxidize things, oxidation means we're taking electrons off of things to feed into the electron transport chain to generate ATP. You and I, and everything that you know and love, and every bacterium that you're going to deal with in the laboratory for the most part, are organotrophs. So they're taking their electrons, as you are right now, from glucose, running them through the glycolysis cycle, through the TCA cycle perhaps, through the electron transport chain. But then there are other microbes, and we touched on them a little bit, and I'm going to touch on them a little more right now, that are defined as rock eaters, litho meaning rock. These are microbes that can use reduced inorganic molecules. So these are unusual things. These are things that we can't do. So as an example of that, we'll talk a little bit about the history of microbiology, although I'm not interested in you understanding the history of microbiology. But Sergei Winogradsky was the father of lithotrophy. And he actually discovered this in the 19th century, which was a really remarkable thing because at this point, every microbiologist in the world, and microbiology was still a relatively new field, was dealing with human pathogens. So they were all dealing with heterotrophs, staph, strep, and the anthrax bacillus. That was what most people were interested in. But Winogradsky, this Russian microbiologist born in Kiev, we just missed his 161st birthday, I'm sorry, it was September 1st. But he was actually so ahead of his time that he was one of the first people who was interested in how microbes that didn't interact with the human host or animals interacted. And so he was the first person to show that there were microbes that could oxidize hydrogen sulfide, that stinky, rotten egg smelling gas that you can smell sometimes in swamps. Well, if you look at this molecule, you can tell that it's a highly reduced molecule. And again, as I think I mentioned in an early lecture, if you think of substituting that sulfur for a carbon, you can imagine that we could digest this and get energy. So the microbes, like Beggiatoa, which he studied, which lives at the interface between the atmosphere and water in microbial mats, has the ability to oxidize hydrogen sulfide as an energy source. It's taking its electrons from H2S, not from a carbohydrate, as you and I are. It can oxidize it to elemental sulfur and then eventually to sulfate. And what you see inside these cells is these little refractile bodies are actually granules of elemental sulfur. They're a waste product of this microbe. So no one actually knew until 1887, which still blows my mind that somebody could come up with this in the 19th century, but Winogradsky did, showed that not all life forms used organic forms for electron sources. So he was pretty brilliant. We've tried, oops, I'm a little ahead of myself. I got a new toy, so I'm still working on it. Bear with me. One slide. Oh, it doesn't. It wants me to skip the Winogradsky column, but I'm stubborn. Okay, I'm going to skip Winogradsky column. Basically, I was just going to tell you that we've tried for years in the laboratory to have students recreate these Winogradsky columns. And it's never worked. I think now that we're in ISC 3 and we actually have windows facing the sun, I think next year I might actually get my nerve back up and try and create this thing that Winogradsky invented to show the stratification of chemolithotrophic microbes in the soil. But if you look at the major microbial nutritional types of microorganisms, there's actually 4 classifications that you can come up with. Most are either photoautotrophs. So here, photolithotrophic autotrophs. So if you look at this, you can break this down on an exam question, right? The light is the energy source. The electron source is coming from an inorganic molecule, and the carbon source is coming from CO2. So light is the energy, inorganic matter is the electron donor, and CO2 is the carbon source. Plants, right? So, but in terms of microbes, we're really talking mostly about algae and cyanobacteria. So most organisms on Earth, and certainly microbes, fall into this category or this category on the bottom. Hemo-organotrophic heterotrophs. We oftentimes just refer to them as heterotrophs because we are heterotrophs. So we get our energy source from chemicals, not from the sun. It's too bad we couldn't actually be photosynthetic, but we're not. We use organic forms of chemicals, and we use an organic carbon source. So really pretty simple. It's what you're used to thinking about. But these other 2 classifications actually ecologically have a tremendous impact on our ecosystem. And so before we kind of go off and talk for most of the rest of the class about microbes that fall into this chemoheterotrophic organotrophic. Let's talk about an ecosystem that is driven by bacteria that fall into this weird group, the chemolithotrophic autotrophs. So these are bacteria that use a chemical source, but they're using an inorganic source. You can tell from the word litho. So the electrons are coming from an inorganic source, and they're using CO2 to build up carbon, or organic carbon. So this is a very different metabolic, physiologic group of microbes. But in certain environments, especially in the deep sea hydrothermal vents, these are the microbes that really drive everything. They are the energy source. Because remember, so now we're going down more than 3,000 meters below the sea surface. So we are way, way below the level that sunlight penetrates. So you and I are used to thinking about ecosystems where plants and algae and such are the primary producers. They do photosynthesis, and then food webs are built on that. Here in the deep sea hydrothermal vent area, there is no photosynthesis. There's a little bit of food slowly coming down, raining down here from the photic layer. But for the most part, the energy that drives this really diverse ecosystem that occurs at these deep sea hydrothermal vents is all based on chemical energy and inorganic energy. So what happens down at these sites? So these sites are fairly restricted, these hydrothermal vents. They tend to occur at sites where the Earth's tectonic plates are either coming apart or they're being driven together and meeting. And so there's a subduction going on. But in either case, these hydrothermal vents occur only at these regions because in those regions, the magma, the molten portion of the core of the Earth, is actually closer to the surface. So what can happen at these particular areas is that under the tremendous atmospheric pressure of all of these thousands of meters of water, so any scuba divers in the audience know that for every 10 meters, you increase by an atmosphere. So the pressure down here at more than 3,000 meters below the surface is crushing. You know, it's like you can't imagine how tough that or how strong that is. It's so strong that it actually forces a large amount of seawater down through cracks in the basalt. And as that water is pressed further and further down, it becomes closer and closer to this magma, and it becomes superheated. And you all remember that if you want to solubilize things in fluid, one of the best ways to get things solubilized is to heat up the water. Sugar and salts are more soluble in hot water than they are in cold water. So what happens as this water becomes superheated, coming in close contact with the magma, all of these minerals, copper, iron, manganese, sulfur, become supersaturated within that water. And because the water is so hot, it tends to rise. And so it comes up and it breaks out of the crust again in what are known as smokers. This looks like this should be a mountain, right? Like it's a volcano. In reality, these are oftentimes just a few inches or a few feet tall. So these aren't gigantic, don't think of mountains down here. But what happens as this superheated water arises, now supersaturated with all of these minerals, and comes into contact with water at the bottom of the ocean, which is almost uniformly at 3 degrees to 4 degrees centigrade, refrigerator temperature. All of those, or many of those minerals, will now precipitate because cold water doesn't hold on to the minerals as well as hot water. So it looks like smoke coming out of these things, but what it is just things like hydrogen sulfide precipitating from the water. Well, that hydrogen sulfide especially becomes a food source for chemolithotrophic autotrophic bacteria. That by itself would be cool. But what's happened is that entire ecosystems of multicellular organisms, usually I think exclusively all invertebrates, will develop into these really beautiful and very elaborate and very diverse and dense ecosystems at these forms. So what's shown here are these Riftia tube worms, which are some of the first things that were studied for this. But there are crabs, there are shrimp, there are a lot of things in there, and they are all dependent on bacteria that can use the hydrogen sulfide that's being precipitated in this water. So here's a video with some, these are black smokers. And so you can see that it literally looks like smoke coming out, but what you're seeing are the minerals precipitating from this. So this was taken from a deep sea submersible called Alvin. This is actually pretty old, back in '93. These hydrothermal vents were actually only discovered not that long ago by a man named Ballard. He's the guy who discovered the Titanic, very, very famous deep sea explorer. But these ecosystems weren't known when I was in college or even when I was in graduate school. But these are the riftia, or the tube worms, that are so well studied. These things can be 2 to 3 feet or up to a meter or so long. These are large organisms. They have calcareous shells that they can alternatively rise up out of, as they are here, and extend their gills. Or, when bothered, they can contract those. So these beautiful red gills are red for exactly the same reason that yours or my blood is red, hemoglobin. So these are gills that are used for exchanging gas with the environment. So you might think, well, that's pretty boring. By the way, don't tell John Allen or Paul Heidemann that I'm teaching zoology and microbiology, because I don't have a license to teach zoology or invertebrate, but I'm going to do it anyway. Here's another video of actually breaking some of these riftia. Sorry for the volume. I like videos. So the way that so these worms have evolved to now take advantage of these chemolithotrophic autotrophic bacteria. These giant worms will now, from that water, transport not only oxygen and not only CO2, because we do that too, right? We take oxygen in, we use hemoglobin to take put oxygen back out. But they're also transporting hydrogen sulfide. Part of the reason why hydrogen sulfide is a poisonous gas to you and I is that it can compete with oxygen at our hemoglobin for binding. And so you can become, this is a toxic gas. But to these worms, it's actually nutritious, but not directly. So what's happening is the blood is picking up oxygen, CO2, and hydrogen sulfide, and that's what's being distributed through its blood system. Within the coelom of this organism, there are a huge number of cells called alternatively trophosomes, or I'll show you in the next slide, they're also referred to as bacteriocytes. I kind of like bacteriocyte better because it tells you that these are cells that are literally packed with bacteria. Endosymbionic bacteria that have the ability to oxidize hydrogen sulfide. So, and I like the term trophosome also because it tells you something about another thing that these cells do. Troph meaning to feed or to eat. These are the cells that generate the food for the worm. So what's happening is that the worm is actually holding these domesticated bacteria inside these bacteriocytes or trophosomes, feeding them hydrogen sulfide, feeding them carbon dioxide, and also nitrate coming in from the water as well. And these bacteria are using those molecules to produce organics. This worm does not have a digestive tract. It used to, evolutionarily, its ancestors had a full mouth and anus, complete gastrointestinal tract, but now they've evolved, they don't need it any longer. They're getting all of their food from exchange from these bacteria, giving organics. It sounds like it's a real friendly kind of endosymbiosis, but really you have to think of these trophosomes kind of as ranches, right? Or a chicken house, or a pigsty. Because not only are these bacteria, these endosymbiotic bacteria, making organics to feed, the host is also able to digest some of the bacteria. So there's a sacrifice going on. That, somebody's being taken to the slaughterhouse quite frequently. So it's not quite as friendly as you might think, but it's a really highly evolved. So this sulfide is coming in along with oxygen. The Calvin cycle is going on here with rubisco, and these reduced carbon molecules are being translocated to the cells. Here's another way to look at this. So this is, in a way, this is kind of a dull image, but I really like this image because if you stop and look at it for a few minutes, you can see that in these concentric squares, starting from the outside, here's seawater. The next square inside of that is the host blood. So here's the exchange of hydrogen sulfide from seawater with the blood binding to hemoglobin. The next square within that is the bacteria site, or the same as the trophosome in the previous slide. Within that bacteria site, so that is a host cell, there is, and this is the final concentric circle or concentric square on the inside. This is the symbiotic bacterium. This is the chemolithotrophic autotroph that lives in here. So hydrogen sulfide is being supplied to the cells, which is being oxidized to generate ATP, which is being used now to drive the Calvin cycle. So that CO2, here's CO2 coming in from the seawater into the host blood and then into the parasite, going in probably to carboxysomes. So Rubisco is being used here to put carbon dioxide onto a 5-carbon molecule, which is then split into 3 carbons. You obviously remember this very well from earlier lectures. But in any case, this is the autotrophy portion of this. This is how the bacterium is taking inorganic carbon dioxide and turning it into carbon, which can then be turned into biomass of the bacterium, some of which is going to be passed into the bacteria site, so to feed that cell, to feed the other cells of the worm. So this is a highly efficient, highly evolved system, which has allowed these worms to evolve to huge size, and they don't even have to eat anything. And this is just one example of a multicellular invertebrate organism that lives in these ecosystems. Crabs have evolved different ways to do it. Mussels, like clams, bivalves, have these have these symbiotic bacteria in their gills. They're not actually eating food. They're all relying on these bacteria. This is actually where many people now believe life began, is at these hydrothermal vents because of the high temperature and all the chemicals present. So this is perhaps believed to be some of the earliest forms of the beginning of life. Now, if you find this even remotely interesting, or if you're interested in marine science at all, you should know, I only found out about this the other day, there is a talk that's going to occur next Wednesday immediately after this class. Unfortunately, it's a rather small classroom, ISC 2280. So if you hurry after this class, please go to this. But Dr. Dijana Figueroa, excuse me if I've mispronounced her name, because I am not a microbiologist. No, I am a microbiologist. I'm not a marine biologist, so I don't know this young woman. But she's here to give a seminar. She's very well known for her deep sea hydrothermal vent research. I don't believe she's a microbiologist, but she can probably tell you, because it's on her introductory slide. Here's that Riftia Pachyptia. So she obviously knows more about this than I do. So if you're either already interested in deep sea hydrothermal vents, or now you're really interested in deep sea hydrothermal vents, please try and get to her talk. I think you'll enjoy it. I'm hoping to get out of class, quickly run over for that. Okay, enough of that. Let's go on now and talk about how it is that microbes take up energy. So now we're largely talking about heterotrophic bacteria. How are they taking up the nutrients? And then what are they going to do with those nutrients? So uptake can occur by a lot of different ways. I'm going to dispense with this pretty quickly because while facilitated diffusion and transport is kind of common in cells like yours and mine, it's very seldom used by bacteria because in facilitated diffusion, there's no energy involved in taking up a carbon source or an energy source from the outside. It just has to do with a concentration difference. So you have a permease molecule like this that can either take up, let's call this a sugar on the outside of the cell and dump it to the inside, but it's just as likely to take one on the inside and drive it out. So this only works if the cells are in a concentration gradient, so with more nutrients on the outside than the inside. Bacteria very seldom, if ever, find themselves in their natural environment in a place where they have more nutrients outside than inside. So this is a system that works well for you and I, but not so much for bacteria. So we're just going to move past that and talk about the active transport of nutrients, which always requires energy. This is because bacteria usually exist in a kind of a challenging environment where nutrients are at relatively low concentration, so they have to invest some energy to actually be able to import them. So we're going to talk about three different classes of transport systems, and this slide shows all three of them, and then we're going to go through them one at a time. So the three classes are either simple transport, shown on the top, where you have a single membrane spanning channel that can use as an energy source the proton motive force. This is the same proton motive force that drives flagellar rotation. So we haven't yet talked about it, but remember that the proton motive force is driven by the electron transport chain. So there's more protons on the outside of the cytoplasmic membrane. So in simple transport, that potential energy of that charge differential can be used. So as that proton passes across, that decreases the potential energy across that membrane. So there's some loss of potential energy, which in this case is used to do work. In this case, to transport a substance, a nutrient, into that cytoplasm. So that's simple transport. Then the next one that we'll talk about in more detail is what's known as group translocation. This is one of the major ways that bacteria transport sugars into their cells. So it's a very important process. This process uses another energy source. It uses ATP or perhaps GTP. Let me back up. It's using PEP, phosphoenolpyruvate. And we'll get into the details of that, but you've all memorized the glycolytic pathway at least 3 times in your educational history. So you at least remember that phosphoenolpyruvate, PEP, is a high energy intermediate in the glycolytic pathway. Well, that's the energy source anyway. It's similar to ATP. It has a high energy phosphoryl hydride bond that gets put onto the transported molecule as an energy source to transport that in. And we'll talk a lot about this group translocation, but you can see that it's different because as opposed to active transport where the molecule that's brought in is unmodified, in group translocation, the molecule that's transported in becomes phosphorylated. So that's an important distinction between them. And then there's the ABC system. Now, this may sound a little bit similar to you because we talked about in the protein secretion lecture a short time ago, we talked about type 1 secretion, which also goes by the term ABC transport. And in that case, we were talking about molecules coming from the cytoplasm and going out. This ABC system is for bringing molecules in. But the two systems are very related. They use very similar proteins. So it's not accidental that the names are the same because, again, the ABC doesn't stand for the fact that there are 3 components to this. It's due to the fact that there is an ATP binding cassette. That's where the ABC comes from, on the cytoplasmic side. So here, actual ATP is hydrolyzed. And that energy is used to bring a molecule across. But now you'll notice that that molecule is not modified. It is not phosphorylated. The third component, which is occurring out here in the paraplasm, or perhaps in the external environment, is a solute-specific binding protein. So for instance, in E. coli loves the disaccharide maltose. If you break down glucose with an enzyme and you get a molecule with glucose bound to glucose, that's maltose. E. coli has a maltose binding protein because it loves it so much. It has a specific protein that binds maltose very avidly. And when it does, it quickly runs to this membrane spanning molecule. And when it docks with that, ATP will be hydrolyzed on the other side, and maltose will be brought into the E. coli, and you can hear the E. coli say yum as it brings it in. So these are the 3 classifications of active transport that we'll talk about in bacteria. So let's first talk about simple transporters. So that was the first of those 3. And now, to make it more complicated, of course, the simple transporters can be broken down into 3 different classifications. So remember, these are all simple transporters. The 3 different ones are antiporters, symporters, and uniporters. So the best example of let's start with a symporter, shown here as a phosphate symporter. All of these molecules are going to use, in essence, the charge differential across that membrane as their energy source. So for this phosphate symporter, it's going to take advantage of the fact that there's a proton motive force, there's a higher concentration of protons on the periplasmic side of the membrane. And so by transporting one hydrogen, one proton across there, there's a decrease in potential energy, which can be used as work. And so phosphate will be co-transported with it. So that's why it's called symport. You have both the proton and the molecule, in this case phosphate, coming across simultaneously in the same direction. Another example here, perhaps a better one, because you've all dealt with the lactose operon, and if you haven't learned the lactose operon from a microbiologist yet, I challenge you, you have not learned the lactose operon. You will not escape this class without having another, even more detailed examination of the lactose pathway, lactose operon. But lactose is also brought in the same manner as the symporter. Uses a proton differential so that a proton and a lactose molecule are both brought in simultaneously. You can have an antiporter. So some molecules need to be taken out of the cell. So in this particular case, sodium is being transported out of the cell at the same time that a proton is coming in. So because they're going in opposite directions, this is referred to as an antiporter. But again, there is a change in the differential charge across that membrane. And the last class of the simple transporters is this uniporter. I know of only one example of a uniporter, and that is this one, the potassium uniporter. You'll notice that, so it's called a uniporter because only one molecule is going across that membrane, not two. Every other one of those, whether it was an antiporter or a symporter, there were two molecules passing the membrane. One of them was a proton. In the uniporter, there's no proton going across. Hence, uniporter. The potassium is going across. So what's the energy source? There's no proton. Well, I think it's simply this. The potassium ion is positively charged itself. So it's actually contributing to that charge differential across the membrane. So as a potassium goes across there, the charge differential across that membrane, that battery becomes less well charged. So it captures that energy as the ability to transport. So this is why I think the only example that I know of is of a uniporter involves a positively charged cation. Yeah, so that's enough of that. In fact, Cece. Yeah.
Does bacteria also have the sodium potassium pump?
So do they also have the sodium potassium pump? Yes, they do. And they also, so many times what they're doing is they're making a sodium gradient out here to then use that to bring in glucose as well. So this sodium, it's not like the sodium is necessarily just toxic to the bacterium, so they're trying to get rid of it. They're actually setting up another gradient that another transport molecule can use. But I think they do have the sodium potassium pump as well, I think. And when we come back on Friday, we'll continue on with the ATP binding cassette transporters. I'll see you guys on Friday.